from Microbe TV, this is TWiP, This Week in Parasitism, episode 242, recorded on August 30th, 2024. I'm Vincent Racaniello, and you're listening to the podcast all about parasites. Joining me today from New York, Daniel Griffin.
Hello, everyone.
The peripatetic Daniel Griffin, back from your travels.
Yes, just late last night, got back from Taiwan. It was sort of funny because I was referring to things that happened on Thursday, and my Thursday was, an extra 12 hours long. So,
are you tired?
I think I've switched over, which is good. I seem to be all right at the back and forth. So
good.
We'll see how that goes. But
welcome back. Also joining us from Glasgow, Scotland, Christina Naula.
Hello, everybody. I've not been traveling, but I walked to Buyers Road today for a coffee.
All right.
It's not as far as Daniel went.
No, it was only about 2.5 kilometers, so nothing much. But, it wasn't raining, so.
Okay.
Yeah, I got to do the longest plane flights in the world, I guess. First I flew to Singapore. That was 20 hours nonstop.
From New York?
Flight back from Taiwan was only 19.
So where did you leave from JFK to Singapore?
So there's a Newark, New Jersey to Singapore flight, which is that 20-hour one. And then my flight back was from Taipei, Taiwan to JFK.
What's the airline from the Newark, Singapore?
It's Singapore Air. And then it was China Airlines.
Wow.
Taiwan Airlines on the way back. So
what do you do when you're on a plane for 20 hours? That just seems like... it's a long time.
It's paradise. I can just read all my scientific articles. I mean, no one bothers me. If anything, they ask me, would you like a coffee or something? Yeah. So it's just,
Imagine they put enough fuel in a plane to go 20 hours,
I
right?
know. Yeah.
It's amazing, actually.
Yeah.
Well, I'm sure you're comfortable up there in the front, so...
Yeah, well, I can read interesting articles like the one that we're going to talk about today.
All right. So if you like these programs, before we jump into our paper, we'd love to have your support to run them. Microbe TV is the production company that makes these podcasts, these science discussions. We depend on your support. We don't do ads. So if you go to microbe.tv/contribute, we have various ways that you can help us. And in the U.S., your contributions are federal tax deductible. So today is paper day. We are going to teach you a little bit of science, and Daniel is going to do the honors.
All right. I'm very, very excited. So, unfortunately, this is not open access. That was sort of a downer, but this was sent to us by Michael Schmidt. And so the article, Intestinal Helminth Infection Impairs Vaccine-Induced T-cell Responses and Protection Against SARS-CoV-2 in Mice, published in Science Translational Medicine. And a number of authors, Pritesh Desai, Courtney E. Carl, Baoling Yi, Chia Yu Liang, Tamara Garcia Salam, Anna Carolina Santana, Felipe Tenkatan, Joseph F. Urban Jr., Seda M. L. Bashir, Darren K. Edwards, Susan P. Ribeiro, Larissa B. Thackey, Rafik P. Sakali, and Michael S. Diamond. And they're from all over the place, right? So we've got Department of Medicine, Wash U St. Louis School of Medicine. We've got Department of Molecular Biology, also Wash U. A bunch of folks here from Wash U, Emory Vaccine Center as well down there in Atlanta. We've got someone from the U.S. Department of Agriculture. Isn't that like if you're a spy, you say you work for the Department of Agriculture? So who knows there? We'll see. We also have got Animal Parasitic Disease Lab in Beltsville, Maryland. Let's see what we've else. Someone from Cambridge, Winship Cancer Institute, more Emory, and more WashU. So people from all around. So quite a collaboration here. And so I was going to start by kind of going through an overview But then, this is, I think, an article that benefits from a little bit of background. So let me sort of give a little bit of an overview. Disappointed Dixon isn't going to be here to talk to us about this intestinal roundworm, but let's go through it. So we start off in I think a nice abstract, which is almost like an overview. So they start by pointing out that vaccines have reduced the burden of COVID-19, but the efficacy in helminth infection endemic areas is not well characterized. And this has sort of come up. What's this potential interaction with helminths and COVID-19 and COVID-19 vaccines? We'll talk a little bit about why that's particularly relevant in a lot of our discussions and sort of discussions overall. So here the group evaluated infection with a murine intestinal roundworm, this Heligmosomoides polygyrus Baccari, right? And they're looking at the impact this has on they say the efficacy of mRNA vaccine targeting the Wuhan 1 spike protein of severe acute respiratory syndrome coronavirus 2 in mice. This is a mouse study. I'll point that out. They say that although immunization generated similar B-cell responses in these worm-infected and uninfected mice, the T-cell responses were markedly reduced in the worm-infected mice. And then they're going to go on and tell us a little bit about maybe some differences with the Omicron variant.
So, Daniel, this Heligmosomoides, what would be the analogous worms for people?
I think it would be a roundworm, a hookworm maybe. It's a roundworm. But
it's intestinal roundworm. So I was thinking Ascaris would be, which makes a lot of sense, right? That's
I've looked it up. Actually, the life cycle seems quite similar to that of Ascaris. So it's only got the single host, the mouse, and unlike Ascaris, it doesn't have a lung transmission. So it's entirely gastrointestinal. So it is transmitted by ingestion of L3 larvae, which seem to be still in the eggs, or at least associated. So they're kind of arrested in that life cycle phase, and the mouse ingests them, and then they develop to adults in the intestinal lumen. And let me just check which part. It just says the lumen of the intestine. So I'm not entirely sure which part of the intestine, maybe all of it. I think it looks like the small bowel, maybe, but I'm not sure. So I wouldn't bet my life on that. And then produces eggs, and eggs are shed, and there's 2 molts within the eggs, and then the life cycle is complete. So quite a simple life cycle, and actually it's quite a pretty looking little worm. If maybe our readers want to put that into Dr. Google,
put a picture in the show notes, maybe.
Yeah, I've got one, so I can copy it in. So I'll do that.
Good looking worm, huh?
Yeah, it is I mean, for a worm.
Well, I think worms like each other,
Yeah. Daniel, I wanted to ask you, because I was wondering, if you're a regular TWiP or immune listener, you probably know about B cells and T cell responses, but I don't know, maybe we should very briefly explain the difference for our parasite enthusiasts.
I'll do it at a high level, and then we're even going to go a little bit into the weeds on this. But as pointed out there, they really feel like this is going to be a model to help us with all the different, I'll say, gastrointestinal worms, right? So, we've got Necator americanus and Ancylostoma duodenale and Ancylostoma Ceylonicum, and also, so our hookworms, right? And then we've also got Ascaris and the whipworm that apparently we love so much we named the same thing twice, Trichurus trichuria. So they're thinking this is going to be a model for that. And Vincent and I have actually talked quite a bit, I think, on TWiP about not only vaccines, but also immune responses and, T cells versus B cells, right? Everyone always cares about B cells and neutralizing antibodies. But we also care about T cells. And there's 2 ways to look at this, right? And I think a lot of times we just simply say, hey, are we seeing neutralizing antibodies? And hey, are we seeing T cells? But there's also this Th1, Th2 paradigm, right? And that was introduced in the 1980s. And that's actually, introduced in the 1980s. And I put a couple links into some of the original Mossman-Kaufman papers where this concept is introduced. And they even have, you could tell it's a 1980s paper because the figure looks like it was hand-drawn, right? Mm-hmm. Remember that? We used to hand draw stuff.
Yep.
And sort of this whole concept that, there's because they're all related, right? Even when we talk about B cells, a lot of times those B cells have T cell help, right? So sometimes instead of just saying T cell, B cell, we talk about a Th1, which has certain signature cytokines like interferon gamma. And then we have Th2, where maybe the signature cytokine is interleukin 4. And that Th2 is going to lead to activation of our B cells and production of our neutralizing antibodies. And I think the point that we've discovered over the years is that you want a balanced response. Right. And this was sort of this idea that if you get too much of a Th2, you don't just help the B cells, but you also get this enhanced respiratory response. You get mucus production, you get eosinophil recruitment, which maybe that's good in an intestinal worm infection, because the what is that? The weep and sweep phenomenon, right? Where you get all this mucus and secretion and hypermotility, and even the enteric nervous system gets fired up, and you try to get rid of those worms. You really don't want that going on in your lungs when you have like a respiratory infection. You want like all this excess mucus and response. And I think that was kind of the Barney Graham concern with maybe that formaldehyde RSV original vaccine, that you got this exaggerated Th2 response in certain individuals. Maybe they had some IL-4 polymorphisms, right? So that was the original sort of 1980s Th1, Th2 idea, because we're going to get into that. But for this paper, you actually have to maybe understand a little bit more about this Th1, Th2. So simple, right? Th1 is a good, solid, cellular type response. A Th2 is going to be that other response where you're going to have T helper cells working with your B cells. You're going to have your signature cytokines. You're even going to have your signature transcription factors, which we're going to get into the different stats. But I think the complexity now we realize is you want a balance, right? You want your T cells, you want your neutralizing antibodies, but there's a little bit of plasticity here. So, in a sense, this model is somewhat dated, but still useful.
So, Daniel, the idea that helminths skew responses is it beneficial to the helminth, or is it a host response adaptation?
Yes.
Both. I guess it's both. Yeah.
Right. I mean, I think that's the thing here is we've co-evolved, right, with the helminths. So, right, you would think, in general, you'd think like this seems bad. Like the Th2 skewing is really the host trying to do what it wants to do, right? Theoretically from a host, I would view this as adaptive. But as we've seen, certain helminths actually need eosinophils. They want to trigger eosinophils, particularly Dixon's favorite, trichinella. If you shut down the eosinophils, trichinella can't form nurse cells. So, this was originally, I think, chicken and the egg concept was originally the way our immune system responded. But then at the same level, I think that the helminths have somehow taken advantages. I mean, if you're an Ascaris female making 200,000 eggs a day, you want those eggs to be expelled out into the environment. So there's a certain advantage to a weep and sweep for the helminth.
Right.
Right. But not too excessive a weep and sweep, because then, he's always swimming upstream against this. All right. So that's our groundwork. That's all you need to know. Now we're ready to plunge.
I've actually got a question.
Okay.
Well, not so much me, but maybe our listeners. So you mentioned earlier a cellular response, that would be Th1. What do you mean by that?
Yeah, it's interesting. So, it is when you say cellular, but Dan, you just Dr. Griffin, you call me Dr. Griffin. You guys can call me Dan. but the listeners, they have to call me Dr. No. We sort of simplify it that way and say, oh, so, the cellular or intracellular, the gamma interferon, this is the kind of response you would think you would want for intracellular microbes like viruses, like tuberculosis, like a few others that you might think of, Legionella. Leishmania, right? Yeah. All our trypanosomes, some of the trypanosomes,
some of them,
some of the trypanosomes, like maybe Chagas, T. cruzi. So, but then extracellular one, antibodies. So, it's not as clean as that. So our interferon gamma signature is going to relate to activation of not only T4 helper cells, but cytotoxic CD8 positive cells. So when we say cellular, it's really almost an intracellular pathogen targeting, where your antibodies, right, are going to code and opsonize those extracellular pathogens.
And then other cells come and gobble them up or destroy them.
Exactly. Yeah. But it is interesting. You kind of want balance is what we've realized. And particularly, and actually the FDA now asks for this. Say, hey, if you've got a new vaccine, you're thinking about giving this to human beings, we want to see that you've got this nice balance. And it is interesting because the disease enhancement historically came from an excessive Th2 signature and also maybe not enough neutralizing antibodies that bind and, antibody dependent enhancement, but you also can have antibody independent enhancement. All right. Okay, so now maybe we're ready. And then we'll get back to this paradigm because there's not only a balance between Th1 and Th2, but you also have to be careful not to induce an over-exuberant T regulatory response, right? Because the T regulatory cells can come in and not so much mess with our B cell antibody production, but they can interfere with that Th1 response, right? So you can end up with an imbalance just because you're driving production of Tregs. And that's going to be relevant in this paper.
And what do Tregs do?
So Tregs, and these, have gone back and forth over time. They do exist. They can end up producing the signature cytokine of IL-10. And then IL-10 can downregulate those CD8 positive cytotoxic T cells. So we think we're all good relevant.
We're good,
Yeah, we think we're all good with our balance, and then the IL-10 downregulates. So, okay. So here's the concern on two levels. So here we're looking at the vaccine response. I mean, the other is how do people do when they actually get an infection? But as you bring up, Vincent, right? Helminth infections tend to skew towards a type 2. So, you go ahead, you do your vaccine studies in, we'll say, a population like the US where there's not a lot of helminth infections. You get this nice Th1, Th2 balance, all good to go. But what happens when you start putting these vaccines into the context of humans, or in this case, mice, that are infected with the helminth? And in this case, they're going to use this model helminth, this murine intestinal roundworm. So going into it, it's like that science thing, the efficacy of helminth infection in the GI tract and COVID-19 vaccine, remains unclear, but here we show, right? So here we're going to go through the data. What are they going to tell us? And it's really interesting. I'm going to walk through the results. I'm going to walk through the figures so that we can all understand these together. But there's a lot of times when they just tell us something and then all the data is in the supplements. And that's going to be okay because some of the stuff in the supplements is going to basically be like, we did a whole bunch of ELISAs, we looked at antibody levels, and this is what we saw. And I don't know if we necessarily need to see all those little dots on a graph. But so they start off in their results section by basically saying, when it comes to the B cell responses, when it comes to looking at those antibody levels, all good here, right? And so they do a prime boost model.
These are mRNA vaccines too, right?
Yeah, so they're going to start off with mRNA vaccines, but don't worry, they're also going to do an adenoviral vector vaccine.
Did any mice get myocarditis, Daniel?
Not in this study. No. Yeah. Unfortunately, mice were harmed. Mice were harmed in the study, but they didn't get myocarditis. So they go ahead and they actually they look at the effect basically on antibodies.
So they immunize and they give the mice worms by mouth, right?
Yeah, well, so they're going to do this in different ways. So they're actually going to look at, as we go through the data, sometimes the mice are like infected ahead of time. Sometimes they're actually, in some of these, they're going to actually treat them with anti-helminth drugs and see how long there's an effect. Sometimes they'll like do the prime and then they'll treat them with the helminth. So they're going to do a couple iterations. And in some of the figures, they actually sort of show you where they're doing this stuff. So
yeah, I thought that was really useful.
Yeah, I like in the figures, right? Each figure starts off with this little time scale. So first they start off and saying it's all good with the B cells, small reductions maybe in the antibody levels, but not a lot to see here. And this is really, you got to go to the supplemental figure for here. And don't worry, when they do the non-mRNA vaccine, they're also going to say, go look at the supplement. So interesting.
See, when they see a small reduction, I'm again speaking as a non-immunologist, so does a small reduction of antibodies not have a significant effect?
No, the question is clinically whether it matters, and we're going to see that in the next experiment, right?
Yeah, I guess.
Yeah, because that is nice. Not only are we going to get some antibody data, not only are we going to get some T cell data, but then at one point they're going to actually challenge the animals, so we're going to see how they do. Okay, so we start off with basically not doing much here on B cells. Go read the supplemental data if you want to see that. They sent us there. But then we get into intestinal helminth infection impairs mRNA vaccine-induced CD8 positive T cell response. So this is we're going to have some impact, some blunted CD8 positive T cells. So this gets us into this wonderfully complicated figure one. I have to say, I don't know how you read this article if you're not like on a nice big computer screen, because the figures are tiny. Yep. And I tried to actually read this, this morning as like a piece of, paper, old fashioned. And I was like, I can't do this. So I had to put it on the big screen to be able to see the plot. So here's this figure. And what they're going to do is they're going to be looking at the CD8 positive T cell responses in mice immunized, right? And so they show us this time scale. So you got the mouse. You're going to do a prime boost. So the prime is day 0. And then 21 days later, you're going to boost with the mRNA vaccine. And then 2 weeks later, you're going to do a post-boost T cell response evaluation. Okay. But then you're also going to have these different iterations. So you're going to have them where you actually can put the worms in, and the worms might be put in before the prime, before the boost. The worms might be put in just before the prime. The worms might be put in just after the prime and before the boost. So they're going to do these different iterations on when they infect the mice. And so going through these figures kind of, P/B. So those are the ones that get it like the prime and boost. Then there's just a prime and there's just a boost, so sort of depending where they get them. It doesn't actually matter, turns out, when we look at the first. So you've got this plot, and I love this nice flow cytometry data. They've got on the y-axis is interferon gamma. On the x-axis is TNF alpha. And what are those? Well, from our preamble earlier, these are signature cytokines of that Th1 response. And you can see here, when you just vaccinate them, no worms involved, you get a really nice interferon gamma, TNF alpha, about 4.6% of the cells in that quadrant. But when you expose them to helminths, really at any point, even before the boost, you basically see a complete blunting of this Th1 response.
That's remarkable.
Really is remarkable. I mean, it's very clear, like black and white. So you get to see the flow, and then they actually go ahead and they graph it out, percent interferon gamma, and just completely blunted. They do another, and so now we're moving into the next. Where apologize for people that don't have open access to this, but the next is we're going to use this modality. We use a tetramer to see, are you making specific T cells, right? So you're going to look for a T cell receptor that's going to be specific. So you're going to use this tetramer. And then they're going to look at CD44, right? So that's going to be a marker of these really murine memory T cells, effector memory, actually, CD44. And they're going to look for effector memory that is going to target, and really you only see it if they're without the helminths, right? So you see a little bit in the helminth, but not much. So 6.25, really nice population. So pretty impressive there. And then again, they quantify that as well. And then they sort of go through it and really look at this in different ways, right? So at the very bottom, they kind of bring it down into this interferon gamma, TNF alpha, and then IL-2. So looking at a lot of these signature cytokines. Okay. All right, so that's going to be our first. That's going to be our first bit of data, where basically they show us pretty impressively that the intestinal helminth is going to impair this Th1 type response. So now they're going to tell us more, and now we're going to look at CD4 positive Th1. So this is really, I think, more of the classic Th1 data. So now we're going to go to Figure 2. Which, of course, I've also blown up because otherwise I can't.
Me too.
And again, you get this really. So we start off with the first plot, right? I think everyone can follow along now. Interferon gamma, TNF alpha. What do you want? You want to see stuff in that double positive quadrant. And you see with just the mRNA vaccine, you get a nice population of our classic. And this is really classic, right? Because this is CD4 positive, which is really our T helper cells. Interferon gamma, TNF alpha, double positive cells. You get a nice population, but you expose these guys to the worms at any point, and basically that population vanishes. Okay. All right. And they sort of walk us through, there's a lot of different ways they're looking at this, and sort of consistent through all the different experiments here. So really kind of, showing us here that, we're getting this really blunted CD4 positive Th1. But here's where you start to get into the meat of things. And this is where they talk about, it's still in the same section of the paper, but the spleens of mice infected with the worms had higher frequencies of these Th2 cells, right? So these are going to be the CD4 positive, FOXP3 negative, GATA3 positive. And so the GATA is actually, what is this GATA, right? And that's going to bring us back to our signature cytokine. So our signature cytokines of our different populations. So Th1, the signature cytokine is T-bet. Th2 is GATA3. So that's our Th2 skewing that we're seeing. And actually, so far, right, B cells seem fine, issues with CD8, issues with CD4. And then Figure 3, we're actually going to see how do the mice do. And so they're actually going to see how do the mice, and we're going to see Figure 3, the worm infection actually is going to impair the mRNA vaccine-mediated protection in an antigenically shifted Omicron variant. So I'm happy they use variant, but don't worry, they're going to use strain too many times going forward. And here you can actually see how the mice do. You're going to basically look at weight, right? So when mice get sick, they lose weight. And at a certain point, there's a certain percent of weight loss that, depending on your facility, you might have to actually euthanize or sacrifice the mice. And so you can see the different outcomes with the mice.
Daniel, here they're looking at two different viruses, don't they? They're looking at an ancestral, yeah, and then in the bottom half they're looking at the Omicron variant. Yeah. And actually, there's a difference there, isn't there? Yeah,
and actually, that's one of the things they make a point of is the difference. So so let's start off with, I think A is the ancestral, right? So so A is ancestral. And you look here, and basically you see ancestral, they're both okay, right?
Yep.
And so here we are. So we're looking at this panel, and what they're doing is they're looking 1, 2, 3, 4, 5 days out, and they're following these mice. And there's 4 conditions. Either they're unvaccinated, which is not great for the mice. Doesn't matter whether you have the worms or not, the outcomes are not great. And then they look at getting the vaccine, whether or not you have the worms, and they actually both do okay.
Yep.
But then they go ahead, and this is interesting, right? I guess everyone keeps celebrating, like, now that we're in the days of Omicron, everything's all better. I mean, I'm really celebrating that we have vaccines, but okay, let's not go there. But here in this model, if they go ahead and they have worm-infected mice, basically the vaccine's not protective.
Yep.
And it's interesting that the antibody response is unaffected. It's the T cell response that is making the mice susceptible, right?
I knew you would love that, Vincent.
I mean,
right?
I love this because this is what we've been saying for 4 years, that it's the T cells that matter. And here this is a beautiful experimental model showing that, you get the antibodies are exactly the same, but if you don't have the right T cells levels, the mice get sick, right?
Yeah. Yeah, look at that. And they do talk about the way, apparently there are several people out there who actually think that the T cell response is what's critical for protecting against disease.
Daniel, as you know, with every variant, the T cell response has been the same, right? It's just the neutralizing antibody response. So why are we chasing variants with boosters every 6 months, right? I don't understand. I mean, this is a peripheral question we could get back to later of why, if you get infected, do a lot of people get sick even though they have the right T cells? Or maybe they don't have the right T cells. Maybe that's the problem with older people, right?
Yep.
But yes, I did love this because it does validate what we've been saying for years, and although in an animal model, it's fine.
That was probably my favorite figure, actually.
Yeah, no, it's a great figure because this is like actually, cut to the chase. Like how do the mice do? Are they protected or not? So, and they go ahead and they show you like the ability of cells to infiltrate. They've got a whole bunch of pathology pictures here. So really great stuff there. But then, we're not done yet because this is, going to go into mechanism. So they're going to start looking at those STAT signaling pathways, right? And so first they're going to look at STAT6. And they're actually not going to see much there. So we're going to move past that because what we're going to really get into is that actually this is IL-10 mediated. So do we remember what IL-10 was? Right? It actually looks like the issue you're getting here is you're going to get Treg cells, and that's actually what's going to interfere with this CD4 positive response with the helminths. They do a bunch of stuff, and I think this is like clinically relevant, where they're treating the mice, trying to get rid of the helminths. And it really looks like even when you get rid of the helminths ahead of time, this Th2 skewing, this impact is persistent.
I found that really interesting, that when the worms are gone, you still have the effect. So how long do you think that would last? It can't be indefinite.
That's the question, right? Like how far ahead of time? Because that's the next series of experiments they need to do. Like, because you might think, we'll just go through, we'll have a deworming campaign ahead of time, and then we'll vaccinate. It's like, how far ahead of time?
Daniel, what fraction of the U.S. population has worms?
A small amount. You have to go to like Alabama, unfortunately.
All right. So this is not an issue in the U.S. at all.
It's not an issue in the U.S., but in a lot of parts of the world, right?
Yeah, of course.
Sub-Saharan Africa. What is it? 1 billion? Like 1 in every 6 people on the planet has an ascaris infection. And then you have, strongyloides and all the hookworms. And
So Daniel, has anyone done a study in areas where there are high worm infestations to look at vaccine efficacy against SARS-CoV-2?
So that's a challenge, actually. As they point out here, basically, we really don't know this answer in humans.
That would be a good clinical study to do.
It'd be a great clinical study. And I think you kind of have to do this level of analysis, right?
Yeah,
so you can't, because what do? We check the antibodies and we say, it's all good here. And they have like this call to action. They in their references, they have a paper. It's kind of an opinion piece. COVID-19 vaccine trials must include helminth infected cohorts in Nature Immunology. Really saying you got to like look at this stuff because you can't just extrapolate from one population and claim that's going to help you out with the other.
And obviously it may also affect other vaccines.
So we didn't talk too much because it's in the supplements, but this actually impacts the what would be a J&J model, so the adenoviral vector vaccine. Which we do these vaccines. The Ebola, one of the Ebola vaccines, is an adenoviral vector. And they didn't really go through, but there is some data that, for instance, the malaria vaccines are impacted by helminths. And so, I think that this is really an important paper for pointing out whenever you're going to do these vaccine efficacy studies, they're not just generalizable to all populations. So if you've got a high prevalence of helminth infection, limited access to clean water, you're going to expect lower efficacy, not just lower efficacy, but you're going to see this skewing towards a, Th2 type response.
So you would say it's worth deworming, as you said before doing vaccine campaigns.
I guess that's the other is if you are going to deworm, how far ahead of time do you have to do that, right? Because this effect looks like it really lasted.
Yeah.
And obviously people get reinfected sometimes quite quickly depending on the circumstances.
That's huge too. Like you have to not only just deworm, but even when we quote unquote deworm, we're a lot of times just reducing the helminth burden, right? Because these go around, take your albendazole once every 3 or 6 months now because of cost issues. Does that really deworm you enough to restore the ability to respond to these vaccines the way someone would without helminth infection at all?
Maybe the key is to design a vaccine that is not influenced by the worm in this way.
Yeah.
I don't know how to do that, but you can imagine, designer vaccines which somehow stimulate a proper T-cell response, even in the presence of the worm influence. Yeah.
Yeah. I mean, it looked like we were okay in this context back, before Omicron, right? At least with the mice, it seemed to, despite, seeing these impacts, it was something about the variant. So I think they do have some studies where they look at, blocking IL-10. So maybe there's some way of blocking this, T suppressor IL-10 mediated blunting. So this is figure 6 where they do this blockade of IL-10 in helminth infected mice restores the CD8 positive T cell response. So I don't know, in a population like this, are you going to have to do something along those lines?
Would you would be a little hesitant to suppress IL-10, wouldn't you, just generally?
I would. I would.
Yeah.
Yes. Yeah.
You'd have to be more specific.
Yeah. I mean, IL-10's there for a reason, right?
Right.
Yeah. So, but this is a great paper. They do all kinds of, I think, really excellent work. I mean, this is not easy work to do. They're doing viral plaque assays, right? The flow cytometry, a lot of this stuff when you're doing intracellular staining, right? You've actually got to fix and permeabilize. And, this is,
So in a broader sense, I like this very much, that the T cells are the ones that are knocked down and make you more susceptible to severe disease. So can we talk about this in people, right? People with every variant wave, I already said this before, but I want to repeat it.
Yeah.
The T cell response is unchanged because the variants don't have any amino acid changes in T cell epitopes that make any difference.
Yeah.
Yet, with each variant wave, people seem to get sick. So if the T cells are still the same, why is that? Is it because these are older people who have poor T cell responses to begin with? Or is it something else?
Yeah, so this is interesting, right? So I'm often, Singapore and Taiwan and talking about COVID and actually some of the questions I get are actually, make you sort of rethink and look closely at the data. So something we've been saying for a while is that over 90% of the hospitalizations, over 90% of the deaths are in people 65 and older, right? 65 and over, chronic medical issues. And there's a whole interesting concept we talk about with hybrid immunity, right? Prior infection, less severe infection going forward. That's true in people under 65. But there was this really interesting data out of Taiwan where they looked at, great data, right? So the last 200,000 people that got infected, and they looked at reinfections, right? And so in general, if you're under 65, reinfection is milder. But if you're over 65, your chance of death actually went up with a reinfection. So there's a difference, right, in your immune system as you get older. Yeah. And a lot of that difference actually is in your T cells.
So does having a matched vaccine to whatever is circulating, does that help?
So it seems to temporarily, right, which is interesting. So, because we always think, oh, well, of course, antibodies go up for a period of time and they come down. Well, T cells actually also can be boosted, their memory jarred, right? So, the data we keep seeing is that if you get a vaccine, the high risk group, right, the folks 65 and over, at least for a period of time, 6 months, let's say 4 to 6 months, you do get a 50% decrease above not getting that booster in your chance of ending up in the hospital, and maybe an even better boost in your protection against death. So, I think we're sort of seeing, and younger people, I don't know, right? Maybe if there's a measles population immunity, it would be great if everyone got those vaccines. But from an individual level, right, that's what sells in America. From, if you're looking out for yourself, if you're 65 or over, then at least for some period of time, and maybe if you get a boost once or twice a year, you can keep that level a little bit lower. And
what is the CDC recommendation? Is it for 65 and up to get the latest booster, or is it just everyone?
So right now, the latest CDC, it's a public health, is that everyone should get it. But again, it's going to be a difference in who gets the most benefit, right? I always go ahead and, get my vaccines all the time because, I am over the age of 50, which is interesting, quadruples my risk of a bad outcome just being over the age of 50.
Yeah. Well, as far as we know, there's no negative of getting.
There is no negative. And, it's like putting on sunscreen before you go out, right? I mean, if I'm in a situation where I'm exposed to the virus and I'm at risk of getting COVID-19, I could reduce my risk of symptomatic COVID in half for 6 months. Yeah. And if I don't have symptomatic COVID, then, maybe I'm less likely to give it to people I care about. So sort of doing my part population-wise, maybe doing a little bit myself. But when you're above 65, then you start getting into, we keep talking about these papers on TWiP where let's say there's already, if you're considered quote unquote high risk, then there's about a 5% chance you might end up in the hospital, 1 in 20. So if you can drop that in half, that's worth doing.
Yeah, I know. So of course there are young people who do have comorbidities, which would make them high risk also, even though they're not over 65, right?
Yeah. So not, let's say you've got, diabetes and cardiac conditions or lung stuff, and you're in your 40s. Yeah. You're still in that group that, ideally everyone runs out, gets their vaccines, and we'll announce later today that Novavax just got approved in the US. But so you got choices.
And then there's also, antivirals have a role to play also, right? So if you were over 65 and you had your booster a few months ago, you're likely to need an antiviral.
Well, I think across the board, right? It's like let's fire on all cylinders. The vaccines are going to reduce your risk of severe disease, and even vaccinated individuals, the antivirals have the same percent reduction, right? So you jump in on day one of a positive test, 90% additional reduction. If you get it somewhere in 5 days, about a 50% reduction. So the antivirals, you start putting everything together. And last year we had 75,000 deaths in the U.S. from COVID-19. I sort of agree with the Surgeon General. The majority of those deaths are preventable with a combination of vaccines and antivirals. Yeah.
So I like this paper because the implication of being the worm burden is clear on vaccine responses, but also it has implications for COVID and COVID vaccination and severe disease. So it hits both.
Yeah, there's a lot. I mean, this is great. And, as they sort of their last sentence, because 1/3 of the global population lives in helminth endemic areas, right? I mean, this stuff's really relevant. So,
Daniel, you mentioned earlier that they were doing some challenging studies as well. Can you remember what that was about?
interesting thing. So they expose them to the virus, but then also, as you could sort of see, they give them the helminths at different times. And really, like, you get the helminth even like after the prime, just even before the boost. That's enough to do it. And then they do these experiments where they're giving them the Pinex or Wormex that we call it, the parental palmoate, basically trying to get rid of the helminths, and doesn't really work. Probably have to get it like so far ahead of time. I guess that's what we still need to do is, they're doing it with or without drug, like 12 or 13 days before the prime. But yeah, it's obviously not enough.
All right. Anything else there, Daniel, you want to mention before we
No,
say goodbye?
I love this. They need to make this open access.
Right.
So everyone can enjoy it as much as us. And I'll leave in links to all these sort of papers that we talked about for background. So the Mossman-Kaufman papers and the O'Shea paper from 2010. So.
And you can also just email the corresponding author to ask for a copy of the PDF.
Yeah, they're usually happy to do that. Yep.
Yes, they are. Yeah,
they're delighted.
Yep.
And I love the image here that was pasted in. Isn't that? I mean, it is a beautiful worm, right?
It is a beautiful worm. Yes.
And it teaches us so much.
Yep.
It kind of looks like one of my house plants, actually, from the Southern Cape that I have in my kitchen.
All right, that's TWiP 242. You can find show notes at microbe.tv/TWiP. You can send questions or comments, twip@microbe.tv. Our next episode will be a clinical case. We're alternating them now, a paper and a clinical case. And so twip@microbe.tv for those guesses. And if you like our work, as I said, please support us, microbe.tv/contribute. Daniel Griffin, Columbia University, Irving Medical Center, parasiteswithoutborders.com. Thank you, Daniel.
Oh, thank you. And everyone be safe.
Christina Nell is at Glasgow University. Thank you, Christina.
Thank you. And thank you, Daniel. You really made that paper very clear.
I'm Vincent Racaniello. You can find me at microbe.tv. I'd like to thank the American Society for Microbiology for their support of TWiP and Ronald Jenkins for the music. You've been listening to This Week in Parasitism. Thanks for joining us. We'll be back next week. Christina.
Another TWiP is parasitic.